So, good evening and welcome to the last week 12 live practice session for the NPTEL course Environmental Biotechnology. I am Guru Priya, a Prime Minister Research Fellow in the Department of Civil Engineering, IIT Madras. This will be our last session, and in today's session, we'll see questions only from the week 12 lecture contents. We have seen questions and concepts from all the previous 11 weeks, and we also had an extra session where we discussed questions from all the previous 11 weeks lectures together last Sunday. In today's session, we'll only focus on the week 12 course content. But before that, can you please tell me if I'm audible and the screen is visible? And also, if you have any specific doubts, kindly post in the chat box so that I can clear that. I'll just wait for a couple of minutes to get these inputs, and after that, I will go about posting the first question for today. Okay, so if you do not have any questions, I will post the first question for today. So this is a match the following question, which is about different techniques, remediation techniques. So you can match the name of the technique with uh, its definition or the components involved, and then please post the answer in the chat box. Doesn't matter if it is right or wrong, just try answering the questions. Okay, so the answer for this question is posted here. So you use membrane technology where it involves semi-permeable barriers and pressure difference as the driving force. So whenever we talk about any of the membrane technology, we are going to have a particular pore size of the membrane, uh, which is going to retain some molecules which are of higher size, and it's only going to allow molecules which are permeable through the membrane. So in remediation process, when we are using this membrane technology, we mostly use semi-permeable membranes that creates barrier for solid substances through pass through. And then, so when there is a barrier, there is obviously a pressure gradient, and through this pressure gradient, the liquid uh, starts moving from one side of the membrane to another. So this is the principle behind membrane technology. When we talk about phytoremediation, phyto stands for plants. So it is basically a plant uh, mediated remediation. So in this, we'll be using plants uh, that grow in contaminated sites, uh, and then they take up the contaminants which we want to remediate. For example, when we talk about metal contamination, we know that metals are not biodegradable but transformable, right? So instead of using microorganisms, we saw a concept of biosorption and bioaccumulation. So similarly, in phytoremediation, it is going to be the root of the plant that is going to absorb and accumulate the metal into the plant structure. And so since the interaction is between the root of the plant and the contaminated site, we call it rhizosphere interaction, and the microorganisms that are present in it are called rhizosphere microbes. Rhizo refers to roots, and rhizosphere microbes are the organisms that are present in the root of the plant. And when we talk about advanced oxidation process, membrane technology is also a physico-chemical process. Phytoremediation is a biological process. Similarly, advanced oxidation process is also a physico-chemical process, and it uses the basic principle is radical generation. So you are going to perform oxidation process by generating some radicals which will oxidize your pollutants. So, and this is also a very fast process. So you use hydrogen peroxide as catalyst, UV as catalyst, uh, and you can you use hydrogen peroxide and UV as the medium, and you use other catalysts which bring about this advanced oxidation process. So it can be electrochemical, it can be photochemical. Advanced oxidation process can be of different types. And biosorption, we have already extensively seen it. Involves passive metabolically independent pollutant removal, because there is no sort of metabolism involved. It's just a adsorption as a surface phenomenon. So like positively charged ions can get sorbed onto the microbial surface, which is usually negatively charged. And then you have anaerobic membrane bioreactor. So this is again a mix of biological and a physicochemical process. So here anaerobic membrane reactor, anaerobic digestion takes place, and for solid liquid separation, you also have a membrane unit in that. So it combines biological and physical filtration, and this process at the end generates biogas. So this is about different techniques that we use in bioremediation. So with this, we will go to the next question. So you have to again match the following, but for each option you can match two options from the right. For primary microplastics, you will match two out of this 1 2 3 4, and with secondary microplastics also you are going to match two options out of this 1 2 3 4. I'll wait for a couple of minutes for you to answer this, and then we will discuss. Is bacteriophage based water treatment considered to be bioremediation? Yes, it is also considered bioremediation technique. So usually we use that to remediate water from bacterial contamination, because phages are uh, virus basically, so they can infect and they can destroy the bacteria. And we also consider phages to be biological agents. So in that case, bacteriophage based water treatment is considered to be bioremediation. Okay. Okay, right. So primary microplastics, they are microbeads that so primary microplastics generally refers to the microbeads that are present in the personal care products, and it also includes plastic fibers from synthetic textiles. Whereas in the case of secondary microplastics, they break down from a larger plastic debris. It's not like directly, it is not a microplastic, it was a bigger plastic. Due to withering, uh, it became microplastic. So that is a secondary microplastic. Whereas primary microplastics in the product itself, it is used in a micro form, a plastic that is used in its micro scale form. And again, secondary microplastics come from breakdown or withering of a larger plastic particle. Right. So the answer is right. Okay. Next, we have a assertion and reason question, again based on microplastics only. Read this and give your answers. Like tell if both are true or either of it is false or both are false. Whether the reason is rightly explaining the assertion. So all this you can take a look at that assertion, reason, think, and then answer. Okay, so the assertion is microplastics are considered major environmental concern because they are easily ingested by marine organisms and then they can enter the food chain. uh, Microplastics are defined, the reason is microplastics are defined as plastic particles less than 5 millimeter in length and are present in products like cosmetics and synthetic clothing. So here both are right, and the reason is explaining the assertion, though it seems a little far. It explains the assertion because uh, only because of the size constraint, uh, the microplastic is easily ingested by the microorganism. That fact is true. And also, this entering into the food chain is also true. Like you have it in cosmetics and synthetic clothing. So it states you from where the source is, and it tells you that it reaches the marine environment, uh, and it reaches the food chain. So it has a very long connection or a distant connection, but still the reason is explaining the assertion here. So the next question is, which enzyme family is primarily involved in the biodegradation of plastics and microplastics? Others, you can also try answering the questions. It is not like whether it is right or wrong, but you can just try. Okay, so the answer for this question is carboxylic ester hydrolysis, because the ester bonds present in the plastics can be broken down by this carboxylic ester hydrolysis. Whereas in the case of amylase, amylase is a starch breaking enzyme. Starch is a carbohydrate basically. And proteases, as the name suggests, it is going to break down protein. And then cellulases is going to break down cellulose. Cellulose is again a carbohydrate. So amylase and cellulase for carbohydrates, proteases for proteins, and carboxylic ester hydrolases are the enzymes that can degrade plastics. Okay. Another kind of simple question relating the pollutant source and the pollutant. So in today's session, we'll see mostly questions of this type only because the last week's course content was also more of pollutants and case studies. So the questions will be very specific and easy. Okay, so the answer is right. Pharmaceuticals arise from human and veterinary medicine, pesticides from agricultural runoff, industrial additives from industrial processes, and algal toxins from they are naturally occurring in water bodies because we do have algae in all lakes, ponds, etc. So again, well, another assertion and reasoning question based on phytoremediation process. Phytoremediation, we saw in the first question that it is a plant-based remediation technique and mostly focused on the rhizosphere region and the microbes present. Okay, so the answer is right. Phytoremediation is economically affordable and non-interfering technology for emerging pollutant removal. That is true because we are just going to grow plants, so it's not economically that costly, and it's also non-interfering, which means that you don't have any secondary effects of carrying out phytoremediation. The reason is it is a rapid process with high removal rates for all type of pollutants. That is false. We know that it takes its own time for a plant to grow and then carry out this remediation. So it is not a rapid and process with high removal rate. It's a slow process, and the removal rate may be higher for certain type of pollutants, but certainly not for all pollutants. It is generally going to be a slower process. Okay, again the type of plastic and its sources. So, these are the things that we see day to day, but are we able to classify the type of plastics that we are able to see? That is the question. exactly matching. So we know PET bottles, we have all this packed juices and all. PVC pipes and polypropylene, we use it for food containers and automotive parts. And polystyrene, we use it for disposable cutlery and for insulating products. Right. Okay, so this is about different techniques and their pros and cons, or different technologies and their pros and cons, probably. Okay. So for membrane technology, it is high cost and energy requirement, because when we talk about membrane technology, the driving force there was the pressure difference. So in order to create that, you have uh, energy requirement, and membrane fouling, membrane clogging is very frequent, so you have to change the membrane frequently, and to get rid of the redundant again, you require cost and energy. So all these factors together uh, cost you a lot of money as well as energy when you go for membrane technology. In case of anaerobic membrane reactor, it is a high energy consuming technique and low efficiency for pharmaceuticals. Any idea why anaerobic membrane reactor has low efficiency for pharmaceuticals? Okay, let me explain the reason again. So, when you talk about pharmaceuticals, pesticides, though they are organic compounds, uh, though we can classify them as organic compounds that are biodegradable, they are highly recalcitrant because of their complex structures. So, that is why anaerobic membrane reactor, which is a combination of both biological as well as physicochemical process, uh, has low efficiency for such compounds. So the low efficiency is not based on the biodegradability itself, but the extent of biodegradability, which you can term it as easily breakable substrate or a recalcitrant substrate. And when you talk about this again phytoremediation, it requires a large land area because you have to grow plants, and the process is going to be very slow. Biosorption, uh, this immobilizing the sorbents is going to be difficult. So with the term itself, you can find out that it's going to match with biosorption. Okay, match these habitats with their characteristics. Today, most of the questions will be match the following, and few questions from assertion and choose the following will be there, just to cover the different concepts. Okay. So when we talk about deep subsurface systems, these systems are largely unexplored, and they have high microbial functional capacity for geological storage. So the term geological storage is going to be the key where we are going to map this particular characteristics with the deep subsurface systems. When we talk about this plant soil ecosystem, obviously there is a term called rhizosphere interaction here. So only out of these four options, only the plant soil ecosystem is going to have this rhizosphere interaction. And photosynthetic carbon fixation, again we map it to plants. In case of marine systems, marine systems contains phytoplankton. So phytoplankton, by the name itself you can identify that we are talking about some kind of a aquatic system, right? So and the only aquatic system that we find here is the marine system. And we also have benthic microalgae, significant dissolved organic matter storage. So all this together we have indications to marine system. And the atmosphere itself, it's a target for carbon removal and not a storage sink. You can't store carbon in the atmosphere. I hope it is clear. If there are any doubts, please post in the chat box so that I'll discuss that again. So here are some techniques regarding carbon dioxide. So match them with the characteristics. Just to cover all the basic concepts at once, we are having more of match the following questions. If it is feeling monotonous, please bear with me for the last session. Okay. So the answer is for enhancing carbon dioxide fixation, we go for improving ATP and reducing power supply for fixation. Whereas when we talk about reducing the carbon dioxide release, when we talk about reducing the carbon dioxide release, we'll be rewiring metabolic pathways, and this is to minimize the decarboxylation. Uh, and then when we talk about engineering energy metabolism, so here we will create alternative carboxylation routes in heterotrophs, whereas when we are trying to build carbon dioxide fixing bypasses, there we go for modifying energy harvesting systems. I think you got confused in engineering energy metabolism and building carbon dioxide fixing bypasses. Like in both the systems, you are trying to modify the system while engineering it for energy metabolism, you are creating carboxylation routes in heterotrophs, whereas for fixing the bypasses, you are modifying the energy harvesting system itself. So here we have carbon capture methods, different methods of carbon capture, and map to some of its characteristics. So identify which of the pair is correct. Out of the four, only one is right, and you have three wrong statements. You have to identify the right pair. Okay, we have one answer, which is deep subsurface sequestration mapped to high investment cost and waste management. Any other answers? So the actual answer is biotechnology and economic feasibility of algal carbon capture, storage and utilization. So when we talk about the physical or chemical carbon capture methods, um, that is going to be mapped to high investment cost and waste management. Because whenever you capture carbon, you have some other product. Again, you have to separate the carbon from it and then convert it into some other product. So this is a loop. So, and in microbial biotechnology, that maps actually with the pair of A, which is low efficiency of microbial carbon dioxide fixation. When you talk about this deep subsurface sequestration, that pairs with leakage in geological storage. So actually, all this A, B, C are juggled. So when we talk about the physical or chemical process, there comes this problem of high investment cost and waste management. In microbial biotechnology, the efficiency is suspected to be low, and in deep subsurface sequestration, you have the problem of leakage in geological storage. Whereas in marine biotechnology, it depends on the economic feasibility of carrying out algae based carbon capture and utilization. Is that okay? Is the answer fine? High investment cost is, although it is true for deep subsurface sequestration, waste management part is more suitable to this physical and chemical carbon capture method rather than for deep subsurface sequestration. We'll move on to the another assertional reasoning question after so much of match the following. Okay, so both of you are telling that both the statements are right, obviously. So the plant soil ecosystem is a major target for carbon sequestration because it has the ability to store large amounts of organic carbon. And that's a good interface to take up the carbon and store it. And then again, it is also easier to bring it into the food chain also. And deep root systems and rhizospheric interactions enhance the stability and longevity of soil organic carbon, obviously. So we have the plants, microbes, all of it that makes this show running. Okay, so from these four statements, identify which statement is false. All the statements are related to carbon capture only. So take your time, read slowly, and identify which of the statement is false. If possible, identify what is false and try correcting it also. So absolutely D is the false statement. So in deep subsurface environments, you have microorganisms that can form biofilms, which is a combination of proteins and carbohydrates. So these biofilms and extracellular polymeric substances uh, generally seal the cracks that form in those environments, and they also help in promoting carbonate precipitation. And despite the second statement states that despite high biomass productivity, there is economic and technological challenges in implementing algal based carbon capture or large scale algal cultivation. That is also true. And the C statement we already saw in one of the match the following question. Optimizing ATP and reducing power supply enhances the rate of carbon dioxide fixation. So this happens in both autotrophic and heterotrophic organisms. So here it is stated that heterotrophic organisms are more efficient at carbon dioxide fixation than autotrophic microorganisms, but it had to be the other way around. Autotrophs are generally more efficient at carbon dioxide fixation than heterotrophs because the carbon source for autotrophs is mostly inorganic carbon, which is carbon dioxide. So they carry out photosynthesis and try to fix this carbon dioxide into an organic carbon. So that is why the potential is high for autotrophs, whereas heterotrophs they do the opposite. They consume the organic products and try to release carbon dioxide. So they are not good at fixing the carbon dioxide as such. Okay, so out of these four, which strategy is most effective for increasing the net carbon dioxide gain in heterotrophic microbial systems? Okay, so mostly the answer was increasing decarboxylation rate. So can any of you explain why it was supposed to be B from your perspective? Any answers? So by enhancing the native carboxylation uh, reactions, we can increase the net carbon dioxide gain in heterotrophs. This is because here we try to improve the activity of carboxylase enzyme and increase the availability of the intracellular carbon dioxide. So this allows more carbon to be fixed into organic compounds rather than the carbon dioxide escaping the system during metabolism. So that is the rationale behind which I gave the answer to be enhancing native carboxylation reactions. So let me repeat. So when we are trying to enhance the native carboxylation reaction, it will increase the net carbon dioxide gain because uh, we are trying to improve the activity of the carboxylase enzyme, and this will increase the availability of the intracellular carbon dioxide. So when we increase the intracellular carbon dioxide, we will have more carbon which could be fixed into organic compounds rather than it getting escaped as carbon dioxide into the atmosphere itself. So that is why enhancing native carboxylation reaction could be a good strategy to increase the carbon dioxide gain in heterotrophic microbial systems. If you have any further clarifications, you can always post in the chat box. If not, if you are convinced with the answer, then it is okay. If you want me to repeat also, please post it in the chat box. So the next question is, in the context of carbon capture, storage and utilization, what is the primary advantage of utilizing marine phytoplanktons and benthic microalgae? Okay, so the answer is they contribute significantly to the global primary productivity and organic matter storage, right? Okay, this question is about microbial fuel cells. The assertion is microbial fuel cells generate electricity without combustion, and in microbial fuel cells, bacteria transfer electrons directly to the cathode, which will then flow through an external circuit to the anode. obviously the assertion is true here. It's just about the reason. Okay, what is wrong with the reason here? Any more answers? So the answer is right. The reason is false. But what should have been uh, changed so that the reason becomes right? It's very simple here. Anyone wants to try? Okay, so the reason is in microbial fuel cells, we will be generating electricity without combustion, and here the bacteria transfers electrons directly to anode, which then flows through an external circuit to the cathode. So that is how the flow is. It's from electron to the anode first and then to the cathode through external circuit. Okay, so this is about first generation and second generation biofuels. So what is the main environmental advantage of second generation ethanol over first generation? What is the difference between a first generation and second generation ethanol? And that will directly end up in the answer here. Okay, right. So first generation biofuels, the feedstock is going to be a food-based feedstock, which can either be maize, sugar, sugarcane, which are maize and sugarcane are mostly used. Apart from that, we have other feedstocks also, like other seeds or grains, all of it. So in second generation, we will not use any food biomass, but instead we'll only use uh, waste biomass. Like in case of sugarcane, the sugarcane remains, so the sugarcane bagasse we use. In case of rice, we can use the husk. So that way, second generation ethanol is preferred over first generation ethanol. So, and then we have another assertion and reason question where the assertion states the functional redundancy in microbial communities enhances the robustness of bioenergy systems. The reason is multiple species can perform similar metabolic functions with this, which will allow the system to withstand the environmental fluctuations. Any more answers for this question, or can we discuss the answer? Okay, so the correct answer is both of the statements are true. So functional redundancy in the sense multiple microorganisms having the same function. For example, when we talk about a simple hydrolysis process, different organisms that can perform hydrolysis or different organisms that can perform fermentation, we will not go for a single species or a pure culture in most environmental studies. This is because functional redundancy is usually beneficial because it increases the robustness of the system. So let's say even though that there is functional redundancy, there may be physical or structural differences in those microorganisms and their level of tolerance for environmental fluctuations is going to be different. Let's say organism A, B, both of them are going to hydrolyze carbohydrates. They are good producers of uh, amylases or cellulases, whatever. So they have good efficiency in performing hydrolysis. On the other hand, organism A is structurally very sensitive to pH changes compared to organism B. In that case, when there is going to be a drastic pH change, if we are going to employ only organism A, then the process is going to be hampered and you again have to do the inoculation start from the first. But instead, when you have different organisms which have similar functions but are physiologically different and have different endurances to different environmental conditions, then you are going to have a greater process stability as a whole when compared to using a pure culture or a single strain organism. So that is the whole point of having functionally redundant microbes with different physiological stages so that they can better handle the environmental fluctuations without actually hampering the efficiency of the process itself. So now you have to match the different types of microorganisms with the products that they can produce. Archaea, methanogenesis, okay, green algae, biodiesel production, okay. Then where do we use geobacter, rhodobacter and cyanobacteria? Right. So, archaea are specially capable of performing methanogenesis, though all the other three steps in this anaerobic process is going to be taken over by bacteria. The key step of methane production is happening mainly due to the archaeal species. So, archaea pairs with methanogenesis, and green algae we go for biodiesel production. Geobacter, again, it's kind of something that revolves around this anaerobic process only. So you have this electron transfer in microbial fuel cells. So geobacter is a exoelectrogen basically. So it can transfer electrons outside its own cell. So geobacter transfers electrons in microbial fuel cells. Rhodobacter helps in biohydrogen production, and cyanobacteria. Cyanobacteria, in the sense, it's going to be pigmented. It's blue-green algae. So we call blue-green algae as cyanobacteria, and with this presence of the pigment itself, we can map it to the photosynthetic energy capture. So we'll be mapping the triggers, microorganisms, or the products to the nature of the reaction that happens. Okay. So talking about the chemical reductant of iron 3, sodium dithionate is actually a strong reducing agent, and it is used to reduce the Fe3 plus to Fe2 plus state. So you can find this in groundwater treatment where sodium dithionate is used to create a reducing environment wherein we convert Fe3 plus to Fe2 plus. And then we have sulfate reducing bacteria which produces sulfide for biomineralization. So you can have hydrogen sulfide being produced in anaerobic process as well. In combination with biogas, you can have methane, carbon dioxide, ammonia as well as H2S. So sulfate reducing bacteria ideally produces sulfide and mineralizes the sulfur from the system. And then you have this geobacter, and I think in from the past 2 to 3 sessions we have been seeing geobacter as a important organism that uh, aids in the reduction of uranium, or uranium remediation happens through geobacter. And then we have polylactate, which stimulates indigenous iron reducing bacteria. And we have uh, iron in its 2 plus oxidation state, which is the abiotic reductant for chromate. So Fe2 plus is not the chemical reductant for Fe3 plus, it's sodium dithionate. Whereas Fe2 plus, it acts as abiotic reductant for chromate. So this matches more, and that will be the right answer. So the next question is, which strategy is most effective for long-term immobilization of uranium in contaminated groundwater? Kind of a question that continues from the previous one. I hope the last answer was satisfactory. If you have any discrepancies, please let me know in the chat box. Otherwise, try to answer this question. Exactly. So, biostimulation with organic carbon sources to activate indigenous iron reducing bacteria is the effective strategy for long term immobilization of uranium. So, chemical reduction is not a long term solution because it involves a lot of cost and again secondary uh, uptake, and all this is going to be costly. Bioaugmentation, again, we don't know if in order to sustain the bioaugmentation also you have to have sufficient nutrient supply. Natural attenuation is also not an effective strategy in case of uranium because it is a heavy metal. Okay. So it is about the different oxidation states of uranium, uranium 4 and 6. Which of it is more mobile and how it behaves. Based on that, you can tell whether the statements are right or wrong. C as in what should I count? Whether the statements are right or wrong. Can you mention? Just keep A as true or false and R as true or false. Okay, right. So both are false. It's simply that uranium 6 is more mobile in groundwater than uranium 4. Again, uranium 6 forms soluble uranyl ions and carbonate complexes, while uranium 4 precipitates as insoluble solids. In both the places, you just swap 4 with 6 and vice versa in all the 4 places, and both the assertion and reason becomes right. Okay, so I think I directly displayed the answer here. So for sulfate reducing bacteria, there is going to be reduction. Iron reducing bacteria, again oxidation of, sorry, reduction of iron 3 to iron 2. Acidophilic chemolithotropes are going to oxidize iron and sulfur both, and Clostridia uh, takes up fermentation of carbon substrates. So with this, we have completed all the questions for today. So just a brief introduction about emerging pollutants. So emerging pollutants, they are um, named so because they are recently identified to be pollutants, and we still don't have regulations for all the emerging pollutants. Let's say we have microplastics, so we don't have regulations for these microorganisms. So regulation in the sense something where we define the permissible limit, acceptable limit, all such things. So we don't have such things for emerging pollutants, and they are defined. They can be synthetic or naturally occurring chemicals, and they are said to have adverse effects on ecology and human health. Right. Uh, and the risks associated with these emerging pollutants include they have a great impact on aquatic ecosystem. Can you please speak quiet for some minutes? Okay. So they have high risk on aquatic ecosystem where they cause this interference with the hormones of both animals and humans. So that is called endocrine disruption. So, endocrine system is the hormonal system in all microorganisms and humans, and these emerging pollutants can cause interference in all these. And it also causes ecosystem disruption because they have this high risk of getting accumulated in the food chain, getting biomagnified, and causing a lot of uh, diseases which are very new. So again, the health sector will also be uh, tormented when we have these pollutants in the food chain. We have to find ways of uh, getting rid of all these pollutants. The health risks can be by direct exposure or through indirect exposure. So it can directly impact the consumption of contaminated drinking water, milk, dairy products, and agricultural products. Indirect exposure will be through secondary sources like agricultural residues and all such stuff. So, and then I have added few slides about anaerobic membrane bioreactors as well. So these two concepts were seemingly important. So whenever I upload the recordings, you can just go through them for your reference. So before closing today's session, if you have any further doubts, you can post now or you can feel free to leave the session. So thank you for joining in for the session. Not only this, for all the previous 12 sessions also, including the extra session, and for your active participation. I hope the session was useful. So if you have any feedback, you can post in the chat box. If you have any questions, you can also post in the chat box. Thank you so much. Any other doubts, or can we close the session for today? Okay, if there are no more doubts, then I think we'll close the session. All the best for your exams and thank you for joining.